so much. Can you hear me? Okay, great. I really want to thank John Mudgett and Tom Fair for inviting me to this conference. As a discovery researcher, I really, in many ways pleasantly not thinking about all the difficult details of making a companion diagnostic. And so this was really kind of giving me a new view and how to look at models as we prepare them and move forward. So I also want to thank Brian for giving such a nice introduction to the model systems, and hopefully he's convinced you that these models can recapitulate immune oncology, and so some of the data I won't even need to go over much. So, how are we using these models? And why? Why would we use them when we have so many great syngeneic models already? Well, at Merck, we really want to enable clinically relevant studies with human cells and understand the tissues. We realize that a lot of the receptors and even some of the markers on specific cells, especially myeloid cells, are different between mouse and human. And especially when it comes to immune oncology, sometimes we are fooled because certain cell types inside the tumor in a mouse may express or not express certain checkpoints. And so it's not a surprise that sometimes we see a beautiful mirroring model and guess wrong. And also, we are thinking that this could give us a really unique, quick sneak peek of how we may go about our biomarker plan with real human cells inside a real tumor that has the microenvironment. So things that we're doing now is we're studying targets that have no rodent ortholog at all. We want to understand cellular mechanisms. We work with PD-1 and CTLA-4 to benchmark, and today I'll show you a couple of examples of how the humanized mouse actually could be compared to mirroring models, and also some work that is in the clinic. And of course, I know that Brian went over well the limitations and lack of HLA and the limited lymph node development. And then there's this residual innate immunity from the mouse that we still have to consider. So I'm going to start by showing some cell line models. And I know that a lot of people do work in the PDX models. And we started with a cell line where we wanted to understand, well, we want to separate the component of wound healing that may happen as you do a surgical procedure with a PDX model, as well as to also not have this variability factor. So we thought to work it out, we will utilize these models. And so when we looked at a melanoma and compared it to a pancreatic tumor, we started to wonder, well, we thought the melanoma would respond to PD-1 and our pancreatic tumor would not, and this would be very clear cut. And it absolutely, just like all immunology experiments, sometimes goes exactly opposite of what you expect. And so what we found in the pancreatic tumor, that we were actually able to have some responses with Keytruda. And in this melanoma model, we did not have very good responses. In fact, almost no responses. And so when we looked more closely at that melanoma, we realized that it was an MSS actually taken from a lymph node of a patient. And so maybe if we had more carefully characterized early on, we would have realized that that would not have responded well to treatment. And then looking at it more closely, we did learn some things about that melanoma. First of all, it all started out that these mice, and very consistent, it doesn't matter which donor, every single donor has done this. Factors produced by that melanoma were causing cachexia and weight loss in these mice, which is relevant for certain tumor types. And along with that, it was a, here's the pointer. We found that we were getting a lot of myeloid cells, and once we put the tumor on board. So this helped us realize that the tumor-produced factors were actually helping to drive the myeloid responses. They egress from the bone marrow where they were already there. There's lots of human myeloid cells inside the bone marrow. They just can't get out because the factors aren't there. But this particular tumor not only drove nice T cell responses and B cell responses. We could see these cells in the periphery and in the spleen, but we were also seeing myeloid cells not only going to the tumor, but also setting up shop in other organs. And then when we compared it to our pancreatic model, which doesn't induce any weight loss at all and has plenty of T cells and seems to have responses, we saw that it was pretty interesting that here we have almost no granulocytic myeloid cells in the tumor and some of the spleen. There's homeostatic reasons why the spleen will not have many granulocytic cells to begin with, but we had more than 10 times the amount of monocytic and granulocytic cells in this melanoma model. And so we realized that that may be a contributing factor. And then also it was a nice surprise that here we have an NSG, base NSG model that we could actually drive myeloid cells and start to understand them. And here is just showing the two different models and how we have the pancreatic, and it had a lot of T cells inside the tumors of these mice, and then when we treat it with anti-PD-1, we more than doubled them, and we have very few T cells inside the tumor of this melanoma, and actually it is almost even though there's plenty of T cells in that mouse, they are being excluded from the tumor. The factors produced by that tumor are making it a cold tumor, and adding PD-1 didn't help. And just showing the signature, we could tell that inside the tumor we did all these gene sets, and we got this really nice T cell signature that was inside the pancreatic tumors, very myeloid-rich signature in the tumors with the melanoma line. So here we concluded that yes, that we can utilize tumor cell lines, and if we understand what they really are making, that they can actually drive biology that we could study further. And that it's very important to understand resistant mechanisms, and we wanted to push that further. So I know that Brian showed some of our work with the GITR model, but I just wanted to show a couple of things that is that here we compared the expression. The Tregs are the blue line, and we have CD4s in the dashed line. And what we could see is that that in the spleen and tumor, that the GITR expression is very similar, and we see that the expression mirrors a lot what we see in human cells, and that when we treat it with the MK4166, and we're able to take down some of that Treg responses, we had nice responses in vivo. But what was very interesting and different than the syngeneic models is that what we see in mouse models is that there is a reduction of Tregs in the tumor and not in the lymph nodes or in the periphery. And we didn't really see that. We didn't see a change in the CD8 to Treg ratios in the humanized mouse, but we saw efficacy. And what we found is that even though the factors produced in that tumor were actually supporting the Tregs, they're still there. However, their expression of their functional markers like ICOS were very high in the control. You see high ICOS, and you see this in human tumors as well. And this was brought way down with the treatment of GITR. So we were actually able to understand IL-2 and interferon gamma responses in the cells that we cultured from these mice, and really say, well, this may give us some insight into when we go look in the clinic. GITR is in the clinical trials now. What will we see? Will it look like the mouse? Will it be more like what we see in a humanized mouse? So that was an interesting one. And so this actually helped us publish this year in Cancer Research. It was a nice publication about understanding how DTA1 and GITR responses actually work. And this is a new molecule that we are interested in. Some targets are very complicated. They hit more than one thing, and sometimes they are on one thing in a mouse are very different in human, and this is one of those where Ceacam it's highly expressed we see on granulocytic cells. And it also has been, let's see, did I skip? Where is my, oh, okay, here, I'll just show this first. It is, here, I'll just do the background. This is a transmembrane glycoprotein, and it's a member of the Ig superfamily. And it's implicated in checkpoint blockade. And early data had suggested that it is expressed on T cells and has a modulatory function. And the characterization in tumors is pretty incomplete. So a lot of work had gone into this. At Merck, we were following this target. And we could see that easily on CD4s and CD8 cells, if you treat with IL-2 or IL-2 plus anti-CD3, that you would upregulate this molecule gangbusters. It would be very highly expressed on T cells. However, inside human tumors and many different tumors, we see the co-expression on granulocytic cells, and we don't really see the expression on T cells inside tumors. So when we went and compared the expression patterns between mouse, like an MC38 model, and mice in a melanoma model versus a human RCC, we could also see that we didn't see much expression. We saw a little bit of expression on Tregs in RCC, but the expression profile on granulocytic and monocytic cells, especially granulocytic cells, was very high and more comparable to human tumors than to the mouse. So we did a model study. We took the drug through the paces and treated it in combination with either Keytruda or with ipilimumab. And we were able to determine that, yes, indeed, on the granulocytic cells, we saw cross-blocking, so we knew that the Ceacam1 was actually bound to the cell. And in these studies, we could see that we could not get any mono or combination therapy with Ceacam1, even though in this model, not only was Ceacam1 expressed by granulocytic cells, it was also expressed by the tumor. And we do see some treatment with IPI, and it appeared to be non-statistically reduced in combination with the molecule. But overall, we were disappointed with the effects. And then a whole host of syngeneic model data and other GEM model data came out, and it looked exactly the same. And so this molecule was already in a clinical trial, which ended up being, did not move forward. So I just wanted to show that we can see beautiful FACS profiles of T cells and monocytic cells in the spleen of these mice, and that we can generally see if there is an activation state. Even if we don't get activation in the tumor, we can see that in the spleen. We'll see enlarged spleens, an increased number of T cells in the spleen, and activation markers on those CD4s and CD8s. Okay. Okay. So as we move forward, we have several targets where we really want to understand. You know, everybody is doing every kind of T cell immunotherapy right now, and we really wanted to see if we can hit resistant tumors. Like we are working on colorectal cancer, and we know that myeloid cells are very important in this. And so in many ways, they drive this immune suppression and the inflammation. They help set up niches for metastases and help promote abnormal angiogenesis. So we wanted to look and see, if we try to do some of the kind of diagnostics that we do on human tumors, what would we see? And so we did this study where we sorted out monocytic and granulocytic myeloid cells, and we were able to get a beautiful profile. And you could see that Ceacam1 was very highly expressed on the granulocytic cells, and that there were other cells that were very highly expressed for other things. So we were able to determined that yes, we could drive these myeloid subsets. And furthermore, when we compared it to our human tumor samples, the expression profile patterns were better matched to these humanized mice than they were with our mouse models, which were highly, just in most of the mouse models, we see a huge upregulation of a granulocytic signature, where in most human and humanized mouse tumors that we've been working with, we see this monocytic population in a more higher ratio, like about 70% monocytic compared to the granulocytic cells. So our goal is to build a model system there where you can really understand human tumor microenvironments. So we've been encouraged by this kind of data. And we were able to take granulocytic and monocytic cells and run some RNA-seq on them. And we may see nice treatment-related effects with one of our molecules. But what the really nice thing is that each one of these is a different donor. And we were able to see the exact same response in all the donors, or very similar responses, which are indicating that, yes, maybe we are hitting that pathway for the drug. So in conclusion, we are thinking about a lot of different things here where we wanted to be able to really have the information that would be relevant to human. And we are running so many clinical trials, 600 clinical trials, that it can be overwhelming. And this could be a strategy to help us answer some difficult questions. So with that, thank you for your attention, and happy to take any questions.
Thank you.